this type of data in Cambridge, and you should start immediately.
Hello, everyone. So I'm going to talk about mining chromatin interactions, and this will kind of summarize everything that you saw today, but from a very computational point of view. So again, all this session and why 3D bioinformatics, because as you've maybe convinced by now, there are more and more evidences on the role of the architecture, 3D architecture of a genome in regulating the function of the cell. So some examples are that people have shown that spatial organization is associated with different diseases and with the disposition of disease-associated aberrations like translocations in leukemia, for example. Chromatin conformations were used as classifiers for disease, specifically for leukemia types and for cell types. Even the drugs, efficacy of drugs that modify chromatins has shown to be related to this spatial organization. And as you've heard now, there is a concept of topologically associating domains and transcription factors. And although there is some debate and ongoing research, there seem to be correlated with different epigenetic marks and gene regulation. So mining chromatin interactions, we have these data that are emerging, high throughput chromosome conformation captures, 3C like HiC, PET, and others, and basically they're allowing us to mine the spatial arrangement of our genome at resolution of from 1 kilobase to 1 megabase. And this means that we can now move on from the one-dimensional browsing and annotation that we usually do with the UCC genome browser to something that looks a bit like that. Okay, so this is a potential 3D genome browser where you can zoom in and zoom out and explore the 3D neighborhood of genes and rotate and maybe add some temporal or functional information. But we're actually not there yet. But we can imagine a future where you can take a virtual tour in the nucleus, okay, and explore the changes that take place during cell differentiation or cell development. But let's take a step back. If we consider this, the pathway in which these data, these 3C data, are becoming knowledge about the 3D architecture, we can think about it as an enzymatic reaction where you have data that are gradually becoming into information and knowledge through the help of tools and algorithms. So we start with paired reads, okay, reads or chromatin fragments that were found together. And they are becoming these contact maps that you've seen in previous presentations. And then you can do visualization and you can do topological analysis, okay, that you've just seen. And then spatial inference, so 3D inference, to try and understand 3D models, and then to integrate them with omics or with temporal information. And on the second column of this table, you can see that there are quite few tools emerging, and these are from the last few years, trying to address the different problems, okay, computational problems that are involved in transforming these data into knowledge. And I'm going to talk about the last two problems because they're key and because we still have a lot of challenges that are not addressed yet or not fully addressed yet. And specifically, I'm going to talk about 3D inference and about data integration. So 3D inference. As you've just heard, the main assumption is that this chromatin contact frequency is a spatial proxy. The things that interact more are more likely to be closer in space. And we have two main approaches. One that says these contact frequencies are distance estimators. Under some model, we can basically convert them into distance estimators. The other approach says, let's look at it as a probabilistic measure, okay? This means that the probability of proximity decreases with contact frequency. So again, if we look into these main approaches in terms of methods, if you take the first approach, okay, looking at it as distance estimators, you do multidimensional scaling or different optimization or simulate annealing like Marty Random just showed you. And the idea is to do metric embedding, okay? So take these distance estimators and create a 3D configuration or an ensemble of 3D configurations. if you take the other approach, you usually try to maximize the likelihood of the configuration, giving these probabilistic measures. And as you see, there are already some tools available, and they differ in the specific machine learning method that they use, but they're basically can categorize into these two classes. So what is important is, again, how do we evaluate these methods or these models? And we can define three main measures to evaluate that. First is what we call reproducibility. And this was mainly used even before the model itself to take different replicates from different experiments that were generated with different restriction enzymes, okay? Where the point is that different restriction enzyme may affect the noise and the bias in the data, okay? And here you basically compare the correlation between replicates, okay, or the model that were generated based on these replicates. The other measure is consistency. It's basically comparing that with fish, okay, with imaging technique that already provided us with some knowledge. And the last one is root mean squared deviation, or END, in addition to that correlation with distances. And usually what you do here is the in silico evaluation. And like you've seen in the previous presentation, the idea that you generate some toy models, okay, you predict 3D structures, and then you compare what you predicted with what you generated, and you see how well your method is performing. So just to illustrate a bit more, this is, for example, comparing two replicates generated with ENCA1 and HINT3 after normalizing or correcting for noise with different methods. And usually what people are trying to do is to increase the correlation between the replicates. So the idea is that if I address the noise good enough, then replicates that come from different restriction enzymes will have high correlation. In terms of consistency, just to explain again why it's really important, there are different studies that show that high C or 3C based models do not always agree with fish, with evidence that's coming from molecular techniques. Okay, so either at very high resolution or at low resolution, there were different studies showing that they don't always agree. And this is a problem. We need to understand why, and either our methods do not work, maybe the data itself captures something else. So just to say something about fish, for those who are not familiar with that, when we talk about fish here, the idea is that we put probes, okay, on genes, and we measure the distance between them. And as opposed to the usual high C or 3C, this is single cell measures, okay, because we can capture to see single cells and measure distances and see the variance, okay, and the idea that once you have labeled, you can apply some segmentation methods and really measure the distances. So just to illustrate, this is a model that we predicted from high C contact frequencies at chromosome 1. And we compared that to fish, okay, fish measurements. And what we wanted to see is not that just that the distances between measured with fish and the distances that we predict are correlated, but also that the trend that these researchers found can be captured with our models. And in this specific case, what you see here in green are highly transcribed genes in chromosome 1, and in red are lowly transcribed genes. And what they found in the original paper, Matheus and Najak, in the original paper, they found that highly transcribed genes are far apart when compared to lowly transcribed genes that are more condensed. And we managed to capture the same trend with 3C-based modeling, which is encouraging. The last measure is again more related to in silico configurations, because again, because fish is low throughput, it means that you can measure the distances only between a very small set of genes, okay? So you need to also think about toy models and how to systematically evaluate that. And basically RMSD, given the model that you generated and the model that you predicted, you want to see how similar they are. So you see here 3 examples, okay? And A is 2 superimposed models that are very similar, so the RMSD will be lower, smaller than compared to 2 superimposed models that are very different. Okay, so the RMSD will increase from A to B and from B to C. And if you do that systematically, what happens is you increase the noise, okay, from some point. If you increase a very, very high level of noise, if very, very randomized, you'll get a very high RMSD, and at some point it won't be significant. So you need to evaluate when your method becomes less reliable, what are the limitations of your modeling approach. Now, if you think about 3D inference, it's a step before we do data integration, because once we have a spatial understanding of what's going on, we can then overlay it with other information. Okay, so we can think about 3 axes. One is space, which means that we're going to put different spatial information from different resolutions or scale together. The other one is time. The idea is that now you take time series approach, okay, and you look what happens to the structure as the cell develops or during disease. And the last axis is function. So this has been a topic of a lot of research because people want to integrate, okay, omics information with structure and understand how structure regulates function, what are the interplay between them. So spatial data integration, if you think about the different sources of information that we have electron microscopy that can tell us something about the shape of the nucleus and its size. And then we have FISH that can tell us about chromosomal territories, their size, where they're located, about genes. Even though it's low throughput, it's some information. And then we have high throughput 3C, high C data. And this allows us to go even further down, okay, and to have higher resolution. And again, we're now reaching a kilobase resolution, and hopefully this will even develop more. So we get a really zoom-in effect if we put all this type of information together. And here the challenge is to link these different scales and get something that is physical, is quantitatively physics. Okay, so to get the different properties and to be able to do this zoom in, if you remember the 3D genome browser that I've shown you before, to do this zoom in action from micrometer to nanometer, and to really understand how things are linked. If you think about temporal data integration, you can take, again, a time series approach. So you can think that you do high C, okay, at different time points over the development of the disease, or you induce some treatment or some change and you see what happens. And up until now, there were only a few studies trying to do that, but they've shown really interesting results. For example, in breast cancer, as a result of estrogen treatment, they've shown that the spatial arrangement actually changes. And you can think of the application of that to drugs and their efficacy and other examples. Finally, we have functional data integration. And here there are 2 types of information. There is a point-wise information, like gene expression and methylation and histone modifications. And even if you're not a 3C person, you might be interested in one of these types of information, and to understand them in their spatial context would be really important to understand what is the mechanism behind that. If you think about relational information like co-expression and co-regulation, overlaying them on the network of interaction or on these contact maps could again highlight patterns and correlations. And of course, the 3 axes that I've told you about require that we think about memory and high-performance computing. Some examples that are already being implemented is the use of GPU platforms, is to try and parallelize all the algorithms and tools that I've shown you before and try to see how they can cope with the mass of information and the different challenges that I've just talked about. Now, in terms of data integration, the key approaches that we could take, and some are now starting to take, is to think about ontologies, okay? So much like we think about Go, okay, to do that for the nucleus and try to relate the different components to each other. Machine learning would be a very important part here in terms of comparison and classification, and thinking about taking a multi-scale approach that was mentioned before. And also visualization. Okay, so going back to the 3D genome browsers and virtual reality, because this will allow people to really understand, feel this data. And just to elaborate about machine learning and the different types of research that are now starting to emerge. So in terms of classifications, there were two very interesting examples. One is using support vector machines. And in this research, they took different leukemia types. And using the conformations of the Hoxha cluster, they were able to really classify subtypes of leukemia based on 3C data. Another paper that just came out is using, again, these 3D models as classifiers of cell types. So you see that there is an emerging trend of using the spatial information or the spatial context in order to better classify diseases or cell types and to complement what we already know. Another issue is about comparison. So let's say that we have 3C or high C data from different cell types, and we see some interaction between gene A and gene B, and another interaction between gene A and gene B in another cell type, and we want to compare. We want to say, are they significantly different? Did something happen here? We can compare the 3D models, but we can also compare the interaction frequencies themselves. And here we adapted the base factor that is used a lot in GWAS for this type of challenge. The last approach is multi-scale analysis, also mentioned by Benjamin, and this is to take a hierarchical approach, so moving from a linear segmentation of the genome to looking at the hierarchy in which genomes unfold and trying to understand at which level, functional level, each scale works. Okay, so for example, maybe there is a scale that is related more to gene expression and another scale that is controlled more by methylation. So trying to think about the hierarchical folding of chromosomes. So again, relating back to the presentation of Syed, we want to extend our view towards new biomarkers that are spatial. Okay, so again, give a spatial context to a lot of things that's going on now with gene expression and methylation. And here, for example, taking this idea that, from the human disease epigenome. So using QTLs, but adding also the spatial context. So you can think that there is, today there is expression QTL and methylation QTL and QTL that's trying to link to the lineage. And maybe we can add here another circle with spatial QTL and deepen our understanding of what's going on. So to summarize, these data that give us knowledge about spatial geometry and position of genes could ultimately give us novel biomarkers and a better way to classify and detect things. And this high throughput 3C data is something worth knowing, even if you're not working with it directly or generating, something that's worth considering and integrating into your research workflow. And from a computational point of view, we need more algorithms, more tools that are also parallelizable, that consider the different omics and the data integration challenges in order to realize the potential of these data. And we're now ready for the panel.
Yes. If there is one question for the speaker, and then I would like to ask the speakers if they like to spend 15 minutes here, and
we could just start with the panel.
could